to the presentation of FluoGuide. We are middle of our warrants exercise period, and we felt that it would be appropriate to make a presentation of FluoGuide. Due to the situation, we cannot do it physically, so that's why we are doing it virtually here today. But at FluoGuide, we maximum the surgical outcome, and we do that by intelligent targeting. Our first focus is cancer surgery, and our pitch is very simple. I mean, the population around the world is 15 million patients every year that have cancer, and 80% of them will need surgery during some time of their treatment. The problem is that the surgeon have very much difficulty to see the cancer and feel the cancer because it's very close to normal tissue, and it's not surprisingly that 50% of the patient that undergoes surgery will have a regrowth of the cancer locally afterwards. And our solution to that is very simple. We have an UPAR-targeted fluorophore that binds to the cancer and light it up so the surgeon can see the cancer, and by that they can remove everything. We term ourselves as a supportive medical technology. We do that because we have the surgeons on the one side and we have the medical equipment manufacturers on the other side, two very powerful groups, and we as a small company combine those two and make them much better to do what they already are doing today, and will thereby have enormous impact. Our first indication is glioblastoma, an aggressive brain cancer, and we have had excellent results with our first product. The market is far bigger than that, in the sense that 3 million patients every year could benefit from our technology. We have patent on our first product, and it's issued in Europe and US. Experienced team, and we are listed it on Spotlight.
Yeah, and as Morten mentioned, our solution is quite simple. We make cancer light up, but how does it really work? I'll try to explain it in this illustration here. It's basically our compound FG1. It's injected into a vein before surgery, and the compound then circulates in the blood, binds only to cancer cells and not surrounding normal cells. And when they sit there, they cannot be seen by the naked eye, but since they have a fluorophore on them, if you turn on a special type of light, you could say a night vision, the tumor will light up. And that's actually what you see in the illustration in the middle. You see a brain tumor that lights up. This guides the surgeon to remove the tumor only and not the healthy tissue, simply by removing what lights up, and until there's no more lightening up. Why is this important? It's important for two reasons. First of all, if you want to cure cancer, you need to remove all of the tumor there. If you leave something behind, it will regrow. The other thing is you don't want to remove healthy tissue, in particular not if we are talking on brain tumors. It might be the center of speech that we remove. So it's very important to remove everything, but only what is needed. And that's what our technology does. If you look on the green dot, it's actually a mouse brain with an implanted human brain tumor. And there you can see FG1 makes that tumor light up, and then you remove what lights up. So we can support that you remove all the cancer the first time. We make the surgeon better at doing what they already are doing. By that we increase chance of survival, because you need to remove everything to improve survival. It actually carries lower cost if you can do more efficient surgery right away, because surgery is very expensive compared to the use of this technology. We reduce anxiety in patient, and we actually make the surgeons be able to do a quicker surgery and feel more safe about what they do. So does this really work? We soon move into patients, as you know, in Q3 expectedly. Yes, we have actually demonstrated this in what we call a mouse clinical trial. So we did everything in a mouse model just as if we want to do it in patients. We showed with a human cancer, in this case a pancreatic cancer, and it's a human cancer implanted into mouse, where we let surgeon operate with just the equipment they use on any patient, including robotic surgery. And then we ask them in white light to do what they normally do, go in, remove the tumor, remove the metastasis. And that's what they did. And then we turned up, beforehand we had given FG1, And then when they said we are done, we turned on the special night vision, and then in half of the cases there was tumor left that lighted up. So in half of the cases, if this had been patients, you would have thought you removed everything, but you hadn't. And FG1 was helping the surgeon to realize that, and if it had been used in the first hand, it would have been a total removal, or in most cases a total removal. So the first indication we go for is glioblastoma. And why is this the case? It's because it's a really severe disease. Almost all patients operated in the standard way will have recurrence, which means the tumor will come back. It's very difficult because the way it spreads in the brain is very tricky. It's difficult to see where it starts and ends. It has one of the poorest outcome for survival. The median survival is only a little bit more than a year, and after 5 years, only 5% will still be alive. There are quite some cases. It's still so rare that it has an orphan drug designation, which regulatory-wise give us some advantages. But you also have to keep in mind that 10% of these patients are children. And if you can improve surgery in children, you can really save a lot of years. So we believe this is particularly important. But it's not the only cancer we are going for. Because if you look here, surgery is still a cornerstone in almost any cancer therapy if you want really to heal the patient. And here you can see the recurrence rate. So you can see glioblastoma recurs in almost all cases, but it also goes for some of the very common cancer. At large, all cancers lumped, it's 50% that will recur. So there's a lot to do, and we can come back to that. It also relates to why it's good to have different ligands targeting UPAR in your armamentarium. Here we can see that UPAR, the target, is very intelligently chosen, we believe, because it's not a one cancer, one target. Our technology can be used broadly across almost all cancers, including some of the most common cancers: breast cancer, colorectal cancer, and lung cancer, head and neck cancer, and pancreatic cancer, and then glioblastoma. That would be our first indication. So we want to go after glioblastoma to some of the high volume cancers as well. And here the different compounds will have some added value.
Now we have started the development of our first product. So for doing that, we first have to show that it preclinically works, and we have several studies that have done that has shown that it works very well in glioblastoma, head and neck, and pancreatic cancer. And we actually just repeated it once more in glioblastoma, showing excellent result. Importantly is also that we can manufacture the product for being able to go into humans. And we both need to do the raw material, and we have done that. We have established a supply chain that can take us until commercial supply. And then the other thing is that we need a formulation, we need the drug product, which is easy to handle, stable, and can be used by the physician in their daily practice. And we have also established a very good formulation that can last for a long time, and we right now have produced it as well. So we have ample of material on the shelf for a long shelf life that actually can be used for patients. The last thing is that we need to demonstrate that it is safe, and all through it's been made of known components that are safe in themselves. We have an obligation to show that they're also safe when you combine it into one molecule. We have done that, we have excellent results there as well, and we have a huge room up to what we need of dose, where we have shown no side effects. So we have a very straightforward and simple case to go into the clinical testing that we will start soon. And that's, I mean, soon we submit here end of second quarter, and we will run the result in third quarter this year. One of the very unique things with FluoGuide is our clinical development. It's a short path to the market, in the sense that we need to demonstrate that the surgeon can see the cancer, just as Andreas explained before on the mice clinical trial. They inject it, they are under white light, they switch on the fluorescent light, they test if they have removed everything, and they have the result. So it will take a few weeks for every patient going into it. And we need 2 studies. The one is the one we do here this summer, which is a phase 1/2 study we do with the purpose of demonstrating safety, but also get proof of concept for the human visualization of the cancer. And then we can go straight into a pivotal study, phase 2/3 study, where we actually prove that it can be seen in the patients. We are a medical regulation we are developed under, but we are imaging agent in that sense. So we have clear endpoint, we have no competition of patients, we have a short clinical trials, and it's a very well-defined route to the market. There's some competitors out there, and we belong to the 2nd generation markers that has active targeting. So that means that we bind to a cancer and we light up the cancer. The previous molecules are converted into a fluorescent product inside the cancer, so they make a kind of cloud around the cancer. The other thing is that it's not visible very deep into the tissue, where our product can be seen 1 to 2 centimeters into the tissue. The previous approved products can see it only 1 millimeter, 2 millimeter into it, and that's actually the difference between life and death. If you can see a cancer that grow 2 centimeters away from the main cancer, being able to find that actually make the difference between life and death for the patient. There's the other company, the other blue squares on the graph here. The competitors are 2 that is in phase 3, but with other cancers. They are either then very specific to one cancer, or they can be used on an unspecific target that can go for both normal cells and cancer cells. And the point here is that FluoGuide have chosen a target that can go for all cancer and very specifically to cancer. In that sense, we can go in for many indications with the same product with a very high specificity. We are an experienced team. You've seen Andreas and myself today. Also, Grete is doing all the preclinical development. We all have brought product to the market before several times. We have invented it, we have developed it, and we have commercialized it. And we have a board with a cross-border expertise that is needed for us to get the full value out of FluoGuide. So the next 12 months, what can you expect there? Right now we are preclinical stage company, but in the next 12 months we'll be a clinical phase 3 stage company. So there will be an enormous development over the next 12 months. And just to mention some of the highlights, we will have several regulatory interactions over the next 12 months. We have the first now nationally, and then we go for Europe and US. We have submission in Q2 for the clinical trial we do this summer. We have the result in Q3. We will prepare studies for other indications. Andreas mentioned UPAR can be used for several different other indications. We will do the commercial scale production, and that is needed for us to start phase 3. And that was when we did the direct issue, that was for us to be able to start that now. So we're ready when we have the result of the first study, we can go straight into a phase 3 study. And then we have a prioritized pipeline of products in targeting UPAR, so we can utilize the full potential that is in this target guiding cancer surgery. Thank you very much for your attention.
Thank you for an interesting presentation. We do have some questions, both from investors that have mailed in their questions, but we will start off by today you announced that you signed an agreement with Rikshospitalet and the University of Copenhagen. Can you tell us about the agreement and what it means to you? I'll start off with you, Morten.
Yes, thank you. Yeah, it's very important because UPAR, the potential to utilize UPAR in guiding cancer surgery have many different aspects, many different indications, many different equipment, many different situations you need to address. And by us have a couple of different products in the field will give us maximum flexibility when we commercialize it and when we partner it. So we can sell one product to one partner, another product to another partner, and we can keep the third one for ourselves. And then additionally also the price differentiation. There's some indication where the optimal price are very high, and then some others are, we're going to say, lower price, but it's more larger volumes. So we can do both a red and a green, and we can get the full commercial potential out of UPAR by having to play around with more products. We don't need a lot, but we need maybe 1 or 2, 3 products.
Why exactly do you mean by you need more than one product binding to UPAR?
Yeah, I can explain that. I mean, more on the technology, it's just as Morten explained. The nice thing about UPAR and what also differentiates us from the competitors is that it's really broadly expressed on all kinds of cancers in essence. So we can go for all these different indications, but we can actually fine-tune the characteristics through our development program. And I can give you one example. FG1, which we use for the brain indication, when that circulates in the blood, then it's excreted through the liver. So the liver will light up a little bit. This doesn't mean anything when you're operating in the brain. But if you want to do a cancer that has liver metastasis or the tumor is very close to the liver, you don't want that background. And therefore, in our pipeline, we have fine-tuned, so we have versions that actually are excreted through the kidneys that not at all get to the liver. So these kind of optimizations and tailoring for the different indications while still being targeted at UPAR makes this portfolio necessary to reach out for all these cancer forms.
Does this affect your budget here in 2020, Morten?
No, it does not. I mean, that's a nice part of it. We are doing a lot of preclinical research on these different compounds. We get all the result of that together with our clinical data on our first product, FG1, and together with all that information, we can, as Andreas said, we can kind of put out a portfolio of 2 or 3, 4 compounds for different indications, different situations, different partnering, and that is the beauty of it. So we just want to secure the rights for the compounds before we start making the selection.
Let's say you get your approval and your product is ready to be sold on the market. What is the strategy here? Will you have your own distribution, or are you looking at licensing partnerships?
Yeah, I mean, glioblastoma is selected as one indication for many reasons. One of the reasons is commercially. And that is because the treatment of glioblastoma is very concentrated. So there are some highly specialized centers that treat it. We can, with our own sales force, meaning 10 people in Europe, 10 people in US, we will be able to serve those centers and be able to commercialize it. So it's when you think of it as a company of our kind, we don't build a company to exit it, but we need to be prepared, of course. If we get an offer that we can sell it, but we cannot be locked up, so we cannot kind of take the next step ourselves. And that's one of the beautiful things, that we can actually take glioblastoma itself, we can demonstrate that we have a relevant product, we have a good sales, and then that would be the exit point, or ideal exit point for a typical medical device acquirer. That would be an optimal acquirer. Doesn't say that we necessarily want to sell it, but we at least have the option to. So yes, we can do it ourselves. And Mr. May, for the other indication, we would need distributors. Breast cancer, for instance, is distributed in many different sites, and that would be distributors in this.
Let's talk a little bit about your financial situation. How does it look right now, and going forward?
Well, it looks fine. I mean, first of all, we have a very low fixed burn rate. So we spend all our proceeds, we spend that on developing our first product into the proof of concept study, and then preparing a phase 3 study. That's what we are doing. So in principle, we can run to end of 21, but then we will not be able to start the phase 3 study. We can prepare it. So so we have a very good position for a long time, and fortunately the corona situation have not affected us. We had paperwork to do in the time now, so we still maintain our time plans.
I will go over to some questions from different investors. And I would start off with you, Andreas. Will you be looking at other indications going forward? And if so, what kind of indications would that be?
Yeah, that's a very good question. We want to help as many cancer patients as possible. And since it's still the backbone, cornerstone in most treatment, in particular if you want really to heal the cancer, is surgery, we basically want to go into most cancer types. Which is the nice thing, because UPAR is actually expressed across these cancers. So we mentioned some of the larger volume cancers. Of course, we tested it in head and neck and pancreatic cancer, but breast and lung cancer is quite obvious next step indications. But it will, what we do is based on the results we get out of our animal testing, and remember still these are human cancers implanted in mice. So all those, as you just saw, we did with pancreatic cancer and other types. We have a full program where we test in all these different cancers to make sure in mouse clinical trial that it's likely to work very well in patients. So I would say broadly in cancer, but of course we have a priority based on what we show on animal data.
To the next question, it's two questions, and it relates to the press release you published today. The first one is the investor is asking, I assume that the new patents refer to possible products that can be used instead of, not in combination with FG01. Is that correct?
Yeah, it's correct. It's not meant that you should use two compounds. That's the nice thing about the technology. It's very simple. You inject it and you use it. But as I discussed a little bit before, it's the fine-tuning and the characteristics, so we can optimize it to the different types of cancers. So it's not like it will substitute FG1 for brain cancer, we will use that. But for other cancers, for instance, if you don't want it to be excreted through the liver, we will use these other variants. And the patent, what the patent really covers is these different variants where we have optimized different characteristics in focusing different cancers covers.
And the next question there, and the follow-up questions, do you see any specific indications where you think that the new products will be better than FG01?
Yeah, I mean, again, it depends on which cancers we use, but we have indications of the next line of cancers we will pursue when we kind of when we have done and shown in brain cancer. It was exactly optimized for this. So breast cancer, lung cancer, we expect this next compounds from the pipeline to be better or optimized for that.
Last question, and I will ask you, Morten, what milestones can we look forward to now in the near future?
I mean, we believe FluoGuide is a very exciting company for a lot of reasons, patients, investors, and also working in it. And it's because we're coming now for a preclinical stage company, and in 12 months we could be a stage 3 clinical stage company. So we take enormous development in a very short time. So, I mean, I mentioned on the last slide, it's regulatory interaction, it's a proof of concept in glioblastoma, it's a pipeline of other indications, other products, and it's a preparedness for the phase 3 with the commercial production and the regulatory interaction that is needed before going into a phase 3 program. So, a lot of very attractive and interesting milestones, both for patients and investors.
I wish you all the luck, and I look forward to follow you at FluoGuide in the upcoming months. Thank you, guys.
Thank you
very much.